today I'm going to share a little bit on a new project that we are going to embark on, which is to explore the next generation sequencing as a method to analyze, to test probiotic products. So in this presentation, I'm going to share a little bit of the overview on the challenges that we face in analyzing probiotic products. And later on, we will actually go a little bit into the very brief introduction into the next generation sequencing, look into the various approaches that we could actually explore for the use of the next generation sequencing in the analysis part. However, I won't be able to have enough time to go into much details, on the methods itself, as in this project, we are going to actually put up, guidelines or best practices that fits, for everyone to use. Hopefully that it's not something that too high of a level, to actually set up the lab for you to analyze probiotic products. Okay, so when we talk about probiotic products, so I guess we do not need this statistic. We do not need to look at this graph to tell all of us that probiotic products is getting more and more attention and the market is growing. So as you could see over here in the global probiotic markets, the compound annual growth rate is actually set at about 9 to 9.7% or close to 10%. So in 4 years time, from 2021 to 2025, you are seeing at least 50% increased in the growth rate of probiotic products globally. So by just having a visit to your market, local market, you will be able to see every year you have got more new probiotic products that are available on the shelf. And these probiotic products come with various forms, so not just in one single strains, but they are increasing in the strains that's available in the product itself. And this definitely is giving a huge challenge to authority, to laboratory, to actually analyze and to make sure that the product actually contains the strains as they claim. So these probiotic products is getting a lot more attention nowadays is because of its health benefits that it confer to human consumptions. So there has been more and more research being conducted to prove that probiotic products, by consuming probiotic products, so you are able to improve your gut health. So not just that it will improve your immune systems, it improve your very immediate, observation, the health benefits, which is the indigestion problem, produces various vitamins and nutrients. There's also been recently more and more research to show that there is a relationship, there is a connection between your gut microbiome with our neurological activities. So there has been a lot more research to show that gut microbiome, so it's important to make sure that you actually would be able to learn well so that you feel happy. So, and a lot more. So it's even showing that there may be connection between gut microbiome with dementia, with also depression. So a lot more research is going on due to these health benefits and claims. So there is a hype in probiotic products. So one of the main challenge or probably the challenges we face over here in probiotic products, there are two main categories. So if we could actually look into it. The first one is actually on the health claims, the health benefit. How do we conduct clinical trials to prove that the product are actually healthy and it will confer its health benefits rather than just a claim without evidence, without science to prove it. So how to conduct it? So this is one part that a lot of people are trying to look into a guidelines, a way that we could actually prove the claims as what is being claimed on the product itself. The second thing that we face, in probiotic products, particularly for authority, is actually how do we ensure that the strains being claimed to produce the health are actually the strains that has been claimed. So it's not just about the genus, it's not just about that it is bifidus bacterium, but the strains itself, because different strains, it confer different health benefits, and that has already been validated with the clinical trial. So in this presentation, I'm not going to look into how to validate the health benefits, but more into the laboratory testing. How do we validate that those are the strains that has been claimed to be included in the product itself? So when we talk about probiotic, products, so probiotics are naturally actually present in a lot of fermented foods. They maybe are added artificially into the food itself, or it's just present in the nature. So we have conducted a survey before that by looking into the presence of LAB, lactic acid bacteria, in foods that present in raw vegetables, in fishes, in meats, in tofu, in your pickled vegetables in the market itself in Malaysia. So we found that on raw vegetable itself, lactic acid bacteria present naturally. So in fact, perhaps when you eat raw vegetables, you find that it's easier to go to toilet. It's not entirely due to the fibers that are present in the raw vegetable itself, but the lactic acid bacteria, which is the probiotics, the naturally occurring probiotics. So that's also how you have kimchi, the fermentation actually do not require you to add in probiotic strains because they are naturally present in the raw ingredient itself. So a lot more industry are producing a lot more probiotic products with artificially added probiotic strains, and a lot of these probiotic strains carry a code. So these are all validated strains that has claimed and proven, scientifically proven, to not able to cause hazard to human, but only health, and with certain health. Certain strains will be able to help in indigestion. Certain strains will help to produce extra vitamin B12. So all of these strains has been validated, and these strains need to be tested. So the lab need to be able to detect not to the species level, but to the strain level to prove that these are the strains that has been added. So all of these benefits depend very much on the type of strains that has been added. As I've mentioned before, the validated strains carry a certain number. And secondly, the dosage of the strains being consumed. So research shows that to be able for these strains to survive, to pass through your stomach, the acid in your stomach that may destroy quite a number of strains, to reach the gut and to have enough number to actually grow and colonize your gut and produce that health benefits, we are looking into at least 1 million cells, 10 to the power of 6 cells, probiotic cells that you need to consume. So to reduce all of these destruction, inactivation in the process, we are looking into at least to have 10 to the power of 8 bacterial cells in the product for the health benefits that you are looking into. And that number is actually very important to make sure that the product that you produce actually confer the benefits as you claim. Thirdly, they must be in living conditions. So these microorganisms, these probiotic strains that has been added in the product must be living. So it means that it's able to grow. And this is one of the greatest challenge that we face in a lot of product. So as survey shows, that a lot of products, do not have the ability to actually protect the probiotic strains in its form, particularly in certain type of product itself. So all of these products that we are seeing today is not just being produced in supplement format, but in various forms. So it could be in a food format, you see it in being added into milk powder. So you see, like, being added in different types of food, health benefits food that contain probiotic strains. You are also seeing, like, these probiotic products, like, being added into different drinks. So, or probably, like, present in yogurt. So all of these products, like, create a great challenge to ensure, like, how your probiotic strains could actually survive and maintain in the product itself. And therefore, in the testing, so it's very important, it gives a challenge also because different forms of food matrices or matrices that carry the probiotic may produce limitation in our testing methods. So when we talk about viability of probiotics, like, we are looking into the survivability because that determines, like, how many living cells we are eating, the dosage, and its living condition. So the probiotic products are very prone, like, to destruction, inactivation by the chemical physical parameters that we look into the different pH in the food itself, the temperature being produced, water activity, whether if salt are present or not present, sugars, hydrogen peroxide, bacteriocins, flavors, colorants, different combinations of strains may also affect, like, the survival of different strains. So it's not just by adding 10 strains together and all of these 10 strains will be growing radially. So one of the strains may inactivate another strains. So all of these need to be tested out. And the production method, how it's being produced, the heat treatment, the incubation temperature, the cooling rate of the product, so the packaging materials, all of these are giving an effect to the survivability of the probiotic strains and the microbiological aspect of it. So what is the technological properties of the strains? Like, different strains may have different survivability rates or inactivation rate. The sensitivity to the temperature and pH are all different. And how do you carry the strains? So do you do encapsulation? Do you not do encapsulation? All of these are going to give an impact to the survivability. And finally, the very, very important step, like, to the industry is that the distribution and the storage condition. So how long the shelf life would be. So if you were to test, like, the product, like, at the beginning of, like, the production, the count will be definitely higher. And after, like, being kept, like, for a certain period of time, you will see that, like, the survivability rates start to reduce. So therefore, a lot of products are being produced, like, with probiotic strains way higher compared to whatever needed to be claimed to counter for the inactivation rate of the probiotic strains. And is it going to be cold storage? So are you going to use low temperature or it's going to be room temperature? When we talk about room temperature in Vietnam, you are seeing, like, seasons, like, right now is more chilly. So compared to summertime, like, the temperature may shoot up to 40 degrees. So these are going to cause, like, the variation in the survivability of your product itself. So this is one of the very preliminary study that we asked you, that is still ongoing, like, that we actually go around and sample some probiotic products, like, available in the market, in Malaysian market. And, in Malaysia, like, it's very warm country. So it's summer all year round, like, with normally, like, the temperature can go up to 33 degrees Celsius. So if during transportation, like, the cold chain is not maintained, so in a lorry itself, like, during transportation, the temperature may shoot up to 40 degree or 42 degree within the, during the transportation. And this is all going to give an impact, like, to the survivability of the product itself. So we compare, like, 3 different types of product, the yogurt drinks, yogurt, and also probiotic supplements, like, in capsule. So these show, like, the different distributions, like, of the product itself. So as you can see, like, we found that in yogurt drinks, like, the probiotic strains are more prone to inactivation, so less protection, as the number of the LAB is actually quite low within the product, as compared to yogurt, which is better, and probiotic supplements. And all of these, of course, like, depend on the different technologies being involved over here, and we are not analyzing into the technology as yet, but just as an overall to show you that the number of LAB would vary. And these would affect the efficiency of the product itself. So the current labeling regulation only require manufacturers to list the total weight of the microorganisms being added in the product itself. And the cellular mass does not consist both do not take into consideration of live and dead bacteria because it's based on biomass. Okay, so but according to what I've already said, and all the recommendation, there should be at least 10 to the power of 6 colony forming unit of live probiotic bacteria at the moment of consumptions, and the biomass just doesn't show this number. That's actually for the benefits of all the consumers. So the current standards basically based very much on the culture-dependent methods, such as the ISO 6887, 15214, 20128, and 27205, and a lot more. So I'm not listing everything because some are actually for the use in dairy product, milk, some are for LAB in normal food and feed, and some actually for the different sample processing methods that we usually use in the lab for analysis of probiotic products. And all of these culture-dependent methods are time-consuming, could be very expensive, if you were to really sit down and calculate the cost that has been included in the testing, so it could be very expensive as it involves isolation and identification at the end of the day if you would like to know what strains are actually present. And it may be inaccurate and inefficient, if the strains are stressed up, injured, they may not be grow readily on your agar plate. So even using a standard method, as you could see. So there's been we have had a lot of experience, or we received complaints from a lot of industry that why is it so that when I send my products to 3 different labs and they are all producing different results, even though they are using the same methods? So that's because when we are dealing with microorganisms, the variation, the error rate may be high, and it depends also very much on how the product is being sampled and the different batches of the sample itself. One of the greatest challenge that we face over here is that the current method may not be suitable for novel products and for new probiotic strains that has been constantly being added to the industrial available strains over here. So new methods need to be constantly being developed to cater for the need. So to test for bifidobacterium and to test for lactic acid bacteria, you need to use 2 different methods. So to test for bacillus, you need to use another method. So it's not just one method that fit for all, but you are looking into various methods for one sample itself. If your product contains more than 2 or 3 different types of strains being added over here. So this is one of the greatest challenge. I'm not going to go very much into detail of all of these different methods that are currently being used over here. So this is one of the most commonly used method when we test for food and animal feeding stuff, using the horizontal method to enumerate for mesophilic lactic acid bacteria. So which is a colony counting method at 30 degree mesophilic LAB. The main purpose I show this is that to show you that it's not I would say a perfect method. There are a lot of limitations involved over here, that there may be presence of psychrophilic and also probably thermophilic LAB, and this is not the method that would be able to detect it. So you may not be able to get psychrophilic microorganisms, and not all LAB bacteria grow on MRS agar at pH 5.7. And some may grow very, very slow. So even though we're using 3 days to do the counting, you're still getting colonies that's just pinpoint colony. Those are the slow grower of LAB. And if you attended the session yesterday on biology, so you know that MRS also allow growth of a lot of yeast and a lot other bacteria that is not specific. So your enumeration may overcount or undercount. So the product itself depends on the form of product. It works well for product that contain only probably 1 strains or 2 strains, 3 strains in supplement, but it may not work that well if you are dealing with food product with high microbiological background. So various type of microorganism may present in the product, then you probably will overcount. So those are all the differences, like, the limitation that we see. So to perform, like, surface inoculation, it's more difficult. It's easier for you to actually pick up the colonies, like, for identification, for later on other type of characterization, but you need to provide aerophilic condition, anaerobic condition or microaerobic condition. So using ball plate, you do not need to, so it's easier to be conducted, but to pick up, like, the colony for testing, it may be more difficult. So all of these are limitation and add-on costs, like, for you to conduct the testing. And this is another method, like, for milk product to enumerate presumptive Lactobacillus acidophilus. So why is it presumptive over here? Because it is not definitive. So whatever you are counting, so they may be Lactobacillus johnsoni, it may be Lactobacillus gasseri, it may be Cryptetus. So because the media do not allow discrimination of all these strains. So that's why whatever you enumerate are just presumptive. So you need more validation after this, like, to make sure that the Lactobacillus acidophilus that you see on the plates are actually confirmed to be Lactobacillus acidophilus. So these are the limitation that we see. So, and this is another method, like, as, like, technology move, so we are trying to move away from culture dependent also. So this is a method based on flow cytometry. So it's a way faster method, easier method to count LAB that allow you to discriminate between leaf and dead bacterial cells that require you to do some staining. However, like, the limitation is still, you will not be able to identify the strains to the bacteria to strains level, but just allow you to enumerate LAB as an alternative, like, to the previous ISO methods that I've mentioned. These are some of the methods that I would like to highlight. The main purpose I highlight all of these is to show that culture-dependent methods, like, enumeration over here, currently being used do not allow us to identify to strains level. So these require you to follow up, pick up the strains, do identification, and do sequencing. So that depends on how many colonies you actually pick up from the plate itself. So if you are seeing, like, products with 10 strains, they claim to have 10 strains, how are you going to do it? So that actually adds on a lot of work and is not efficient. So the challenges we face over here, there are three. The type of strains. So how to detect and isolate various strains in product with multiple probiotic strains. If the product only has one strain, no problem. But we are seeing today in the market, the highest number of strains being claimed to present are 21 strains being added over here. How are we going to validate that there are exactly 21 strains present, not more, not less, not 22 strains, not 20 strains? How do we do that? So how to identify correctly to the strain level, not just the genus. I do not want to know just Bifidobacterium. I do not want to know just to the species level. I want to know to their strain level. Okay? So the dosage in living condition, how to effectively enumerate different genus and strains, product with 10 strains. How do I enumerate each strains that it contains how many cells? So how to differentiate between live and dead cells? So it's not just between biomass, but live and dead. And the safety issue. So how to ensure that no other unwanted strain present in the product? How to ensure that there's no mutation actually occur in the strains that you claim to present over there? How do you ensure that there's no other Salmonella, Listeria, Bacillus cereus that you do not want to see that may cause problem to human consumption being actually present in the product itself? So we are seeing these challenges more and more, particularly in the complexity in the products that is being produced in the market itself, particularly when it is consumed as a food or being added in a type of food, like, it probably could be in fermented vegetables, it could be in a fermented milk products, it could be in other fermented meat products that contain all of these strains. So therefore, one of the methods that we look into or currently looking into is the next generation based, like, microbial assay that probably would allow us to look into all these issue that we face over here. There are many platforms available, not just Illumina, there are PacBio, there are nanopores, Oxford nanopores. So a lot of all of these platforms are all forming this next generation sequencing platform that allow you to do parallel sequencing in a very rapid method. Of course, the limitation that we are seeing over here is that it's expensive to actually set up the lab, and you need to train experts to be able to run and analyze, interpret the data itself. So this is just one overview of the potential application of next generation sequencing. So you could use next generation sequencing to look at the microbial population in total. So this is something that we are very interested to look into when we are looking into the probiotic products. So there are two types of application when we look into the microbial population. You could either use the meta barcoding, or some people call it amplicon sequencing, or targeted metagenomic methods that you actually amplify using PCR to amplify the targeted amplicon that you would like to look into, and only after that you subject these amplicon to the next generation sequencing platform that you use for the sequencing. Then the sequences will come out and go into the bioinformatic platform that you need to do computer work. Analyze, look at all the presence of the different genes and align them to the database, and that will tell you that you have 20% of lactobacillus, you have 10% of bifidobacterium, you have this, you have that. Another method is actually using metagenomic. Metagenomic do not require the extra step of amplification of the targeted amplicon, but rather the whole genomic being extracted are being subjected to NGS platform, and sequencing will be done to all the genomic that actually present in the sample. Then follow up with the bioinformatic pipeline that you do analysis of all the sequences present and you fish out so the bacteria sequences that you want, and you do counting and enumeration. So what are when we compare these two methods has been published and being used in the analysis. So we have tried both methods in the analysis itself. So both methods, there are pros and cons. So as you could see over here, the meta barcoding or the targeted metagenomic is cheaper to be run. So because you can run more sample as a PCR steps has been included to amplify the amplicon before actually you do the sequencing. So the bioinformatic process is less complicated, I would say, because you do not need to clean out all the unnecessary data, unnecessary genes that you do not want to look at, but just the amplicon itself. However, using meta barcoding, it's more prone to biases, because whatever happened during the PCR, there may be biases occur, and it may not give you a real composition of the strains present in your sample itself. So you may miss out one or two. So it may not allow identification to strain level, depend on the amplicon that you use. So it could be if you are using 16S, so it probably will only allow you identification to the species level, but not strain level. So this is the limitation of meta barcoding. Although it's easier to be run, it's cheaper to be run. Shotgun metagenomic, on another hand, is costly. So because it's a huge sample, so per package, per run, you can run less sample, and therefore it's more expensive to run. More extensive bioinformatics, in your pipeline. So however, it allows quantification, it allows identification to strain level, and it produces more accurate data on the composition of the present, means that you can count your bacterial cells present in the sample itself. And it also produces more information and data, including all the functional genes. What are these bacteria doing in the product itself, what genes it contains, and so on and so forth. So both of these methods are being used currently. So this is the project that has been approved by APEC, and we are running the project right now in collaboration with AOAC and US FDA. We are currently in the process of forming the expert teams to actually produce guidelines and best practices. So we want to produce recommendation and guidelines for verification of probiotic strains, cell viability and contamination using NGS technology by looking into the various methods that is available, and to build capacity in performing and applying NGS for probiotic testing. So this will happen next year in about May and June time. So the training will be in Malaysia. So, and also very important to initiate a supporting network that consists of few experts, food safety authorities, researchers, industry in APEC to enable communication and continuously improving the methods as we see. So this is a work plan that I'm not going to go into detail, but what I would like to highlight is that we will have a 3-day capacity building workshop next year in Kuala Lumpur, and it will go by invitation. That will include also commercial labs and authority. And finally, we hope, with this project, ongoing, we will be able to better understand, the current status of the lab capacity in APEC, probably, globally, and what works, for all of these, laboratory, including, developing countries. Best practices guidelines, so that we have one standard method. Hopefully, this will lead to the development of a standard method that could fit for use for everyone, and increase, knowledge and lab skill to initiate and kickstart, effort to standardize lab method, increase cooperation and collaboration network, which is one of the most important thing. We are still in a very early beginning stage, and hopefully this will actually kickstart a new era in probiotic testing. With that, I would like to end my presentation, and thank you.